looking at DNA methylation here, looking at the attachment of a methyl group, which is a CH3 component, to a base pair of DNA. And you can see it takes on many different shapes here, but methylation is referring to the attachment of this methyl group. That's what we call methylated. So a methylated DNA molecule. DNA methylation is important for regulating gene expression. This is its main purpose here. There's no change in the sequence, which is an important aspect to keep in mind here, that we're not changing the sequence, but we're still having a way of regulating gene expression. Cannot be used to prevent genes from being expressed. So we're looking at these methyl groups being attached here and indicated by these kind of glowing areas on the DNA molecule. So during gametogenesis, certain genes are methylated and tagged as either maternal or paternal. So how does your body know which one comes from mom, which one comes from dad? It can, in some way, be related to the amount of methylation that occurs. Developing embryos read these tags and express one version over the other. So genes that can be expressed, here's our promoter region, indicating the gene can be expressed. Here's our methyl groups. Methyl groups can cause genes to be inactivated. In this case, there's nothing wrong here. There's no mutations that occurred, but they're inactivated because of the methyl groups attached here, kind of messing up that promoter region, not allowing those genes to be made. So methylation typically found in what's called CpG Islands. C and G you should be familiar with. Those are cytosine and guanine. So these cytosine and guanine-rich areas are located upstream of the promoter region. Of our promoter region, here's our CpG Islands. CpG Islands. The P indicates it's a phosphodiester bond. So in humans, methylation is the result of DNA methyltransferase. So again, it's an enzyme attaching these methyl groups that's responsible for the initial pattern during early development and for copying the pattern during DNA replication. So not only does this occur initially, it can also be replicated when that DNA goes through that process. And our methyl groups typically represented by these dark circles, are unmethylated groups by these open circles, or these white kind of circles here. So this phosphodiester bond, not to get into too much detail, but what this is it's between the nucleotides here, and it's which presents thymine and 2 molecules of adenine. So here's our thymine, and here's our 2 molecules of adenine. This phosphodiester bond is this what we're looking at right here. PO4 to the -3. In this case, we can see right here. Our thymine and 2 molecules of adenine is our phosphodiester bond that's occurring. Now, clone mice with different DNA methylation. So genetically identical individuals can look or behave very differently based on the methylation. Remember, it's affecting the gene expression, not the sequence. Here we have our 2 mice. They look slightly different, but if you look at the tails, there's definitely a difference in the tails. See a nice normal tail here, and this one looks like it got run over or something. But this is because of the methyl groups located in the tail region of this mouse are altering the gene expression. So DNA methylation and probe design. So after a bisulfite treatment, which is just a scientific treatment here, we're trying to determine what was methylated and what was not. The methyl group will protect the cytosine, otherwise would be converted to uracil. So this methyl group, when you have this bisulfite treatment, what's happening with cytosine, it would be converted to uracil under normal conditions. That methyl group, you think of it as being like a block. It's like preventing that bisulfite treatment from changing that. As a result, cytosine is conserved after the treatment. Then you would add a probe, and you would see if it would bind in light. In the case of the methylated probe, that G is going to bind with that C, indicating, oh, light's going to be given off. In the case if it was unmethylated, if this unmethylated probe was added, you can see this has nothing to bind to. This A is not going to bind to that C, it's just going to be floating around. In this case it binds, and again the original strain here was unmethylated. So this methylation probe design is a way to determine through a bisulfite treatment whether or not these methyl groups were present. Again, if it was present, it's going to prevent that cytosine from being changed to a uracil. That's just the result of a typical bisulfite treatment. That methyl group will block that and prevent that from occurring. We use a probe here to determine and indicate whether that was present or not. Something called a COBRA, Combined Bisulfite Restriction Analysis, is all it stands for. Typically used in microbiology labs. It's a test to determine DNA methylation levels. Short general explanation in the following terms. PCR, which we'll learn about in another video, is a quick way to multiply DNA segments, and restriction enzymes cut DNA at very specific locations. So when we have our groups here with the little M's, the bisulfite treatment occurs. The C's stay C's because the methyl group blocks that, and we use our restriction enzyme digestion, and it breaks, in this case, the DNA sequence. And here it's unprotected. Those methyl groups are not present. During the bisulfite treatment, those C's are converted to U's, which then are transferred to T's in DNA, and then we have a restriction enzyme that will not recognize the particular sequence. So this COBRA, you may have heard this term, COBRA is combined bisulfite restriction analysis. It's how we determine the DNA methylation levels. And what this is used for is comparing normal cells to cancer cells. And notice the cancer cells have elevated these levels of methylation. So doesn't mean that if it's methylated, it's cancer. That's not the case. Methylation levels are typically increased in cancer cells here, and that's evident. You can see our cancer cells, a lot more peaks of methylation levels than in our normal cells here. So just one way methylation can help change or regulate gene expression, typically located upstream of a promoter region.